Now, moving on to non-disjunction or abnormal meiosis and the consequences of abnormal meiosis. Remember, during anaphase, both anaphase 1 and anaphase 2, the chromosomes have to separate, right? They have to be pulled to the opposite poles to form the new cells. This is not always the case. Sometimes the chromosomes or the chromatids fail to separate, right? Remember, that is called non-disjunction. Now, if non-disjunction occurs on chromosome pair number 21 in humans, that individual would then have Down syndrome, right? So let's say the female gametes have failed to, there has been that failure to separate on chromosome pair number 21. It means that on chromosome pair number 21, instead of having 2 or 23 chromosomes, we're going to have 23 plus 1 because there's an extra chromosome, right? On pair number 21, instead of having 1, you're now going to have 1 plus 1 because of the extra, right? Now, because that cell would now be abnormal and it is then going to be fertilized by a normal sperm, it means that you are going to have 46 chromosomes, 23 normal from the sperm and 24 from the ovum. We're going to have 46 plus 1. So there is that extra chromosome, and we say that that leads to Down syndrome. So when an abnormal gamete with 24 chromosomes fuses with a normal gamete with 23 chromosomes, then that is how Down syndrome would occur. Remember, Down syndrome only occurs if non-disjunction takes place on chromosome pair number 21 in humans. If the non-disjunction takes place elsewhere, it is not going to lead to Down syndrome. Okay. Now, let's have a look at another activity based on meiosis. It says the diagrams below show a DNA molecule and a phase in meiosis. Diagram 1, as we can see, we have a DNA molecule, and in diagram 2, we can see that we are looking at a phase during meiosis, right? So let's have a look at the labels. As we can see, here we have a chromatid, or we can say this is a chromosome, and then a chromatid, and then at B would be our chromosome. At C, that would be where we have our centriole or centrosomes, and then we have the spindle fibers at D, and we also have another chromosome at E. Right, so just a few minutes for you to go through these questions, and then we are going to quickly discuss them. All right, let's have a look at 2.1.1. The question says, which phase of meiosis is represented in diagram 2? Right, so as we can see there, it is prophase 1. We can see full chromosomes, they are still whole chromosomes. We can also see that we are starting to see the movement of the centrosomes to the opposite poles and the formation of the spindle fibers. Right, okay, so that is prophase 1. Right, and then 2.1.2, how many chromatids have identical DNA from the black shaded chromosomes in diagram 2? Now let's have a look at the chromatids that have identical DNA, looking at the shaded black ones. Okay, so as we can see, we have these two that are the same, we have these two that are also the same, and we have those two that are the same thing. So it means that we are going to have 6 that have identical DNA, because we have 3 chromosomes that are the same. Therefore, 6 chromatids would be identical. Okay, 2.1.3. Describe the composition of the nucleotide in diagram 1, meaning that you are going to describe the composition of the nucleotide for a DNA molecule. Now, we looked at the structure of the nucleotide earlier. You simply describe what that structure is made up of. The nucleotide is made up of a sugar. Remember, the sugar in DNA is a deoxyribose sugar. Now, just a reminder, the sugar in RNA would be a ribose sugar. So it is made up of a deoxyribose sugar. It is made up of a phosphate and a nitrogenous base, right? So DNA is made up, the DNA nucleotide is made up of deoxyribose sugar, a phosphate, and a nitrogenous base. So any of these two would give you those 2 marks. 2.1.4, name and explain the significance of the process occurring in DNA during interphase that will lead to the chromosomes appearance in diagram 2. Now, what process is occurring in interphase? By simply knowing which process occurs in interphase, you will then be able to describe that. Remember, the process that happens during interphase is DNA replication. So, compulsory mark, DNA replication. If you do not name DNA replication, you automatically lose that 1 mark. So now, you are going to describe the significance. You're not describing the process here. You're explaining the significance. So, what is the importance of DNA replication? We know that here, chromosomes which were single threads are going to become double, right? And each chromosome will now consist of 2 chromatids that are joined by the centromere, doubling the genetic material. So the doubling of the genetic material is the importance here. The threads becoming double again is another importance. Remember, this is so that the cells can be able to share the genetic material at the end of the cell division. 2.1.5, explain the role of part D in metaphase of meiosis. Metaphase 1 of meiosis. Now, part D are the spindle fibers. Let's just quickly go back. Spindle fibers, there we have them. So what is the importance of the spindle fibers during metaphase 1? It means now you must know what happens during metaphase. That is the pulling of the chromosome. That is the aligning of the chromosomes at the equator. So it makes sure that the chromosomes are aligned at the equator, right? So the spindle fibers are going to attach to the homologous chromosomes at the equator of the cell, and it is going to lead to those chromosomes being randomly arranged, the random arrangement of the homologous chromosomes, and that will lead to new combinations, or it will lead to genetic variation. Now, if the question was asked in terms of anaphase, you would then mention that during anaphase, the spindle fibers contract or shorten, and as they contract, they are going to pull the chromosomes to the opposite poles. Okay, now let's move on to genetics and inheritance. Here we are going to start by looking at Mendel's laws of inheritance. When we are looking at these laws of inheritance, they are going to be what guides you through the whole chapter, right? As you are looking at Mendel's law of dominance, you're going to be looking at the types of dominance, and you're going to be looking at how dominance basically works, right? The segregation law, the law of independent assortment. Let's have a look at that. So firstly, the law of dominance. When 2 homologous organisms, remember, I said, know your terminologies, right? Now, what do they mean when they say homologous? They mean organisms that have, for example, this would be homologous, this would be homologous as well, because these alleles are the same, right? And then heterozygous, homozygous, and heterozygous. This would be heterozygous. This is hetero, and this is homozygous. Now, when 2 homozygous organisms with contrasting characteristics are crossed, all the individuals of the F1 generation will display the dominant trait. Again, what is dominant? Go back and check what is the dominant trait. That is the trait that is going to show up even when we have a heterozygous genotype. So here, this can represent long hair, and the small letter H represents short hair. So if an individual with long hair is crossed with an individual with short hair, the F1 offspring, the first generation of offspring, will display the dominant trait. Dominant is represented by the capital letter. So it means it will display long hair, right? And these individuals would be heterozygous. However, the dominant one is going to mask the recessive one. It is going to be stronger than the recessive one, and it's going to show up in the physical appearance. Okay, now, in the Mendel's law of segregation, an organism possesses two factors which separate or segregate so that each gamete contains only one of these factors. Now, you will see that as we are looking at genetic crosses, you would see that, let's say we have your genotype. In the genotype, we have H, for example. Now, we have two factors here. We have the long hair and the short hair factor. These two factors are going to separate so that the gamete that is going to be formed, is now going to have only one of these factors. We're not going to have this gamete and this gamete. We show the separation by putting a comma or circling around each of those. So the factors are going to separate during gamete formation, basically. Okay, then the law of independent assortment. The factors controlling the different characteristics, these are the factors, are separate entities, right? And do not influence each other in any way. And these factors sort themselves out independently during gamete formation. We can see that they have separated from each other, right? Now, what this means is that they are separate entities. It simply means that when we are crossing these gametes of this individual with these gametes of the other individual, the two gametes from individual one are not going to both cross with the gamete from the other individual. So we do not have H. This H is going to be independent of the second H, and it's going to independently cross with the H of the second individual. So they do not influence each other because they are separate or independent of each other. It doesn't mean that this H must always pair with these two H's, or these two, or this one must always pair with these two. This has a relationship with this H independently, and it also has a relationship with the other H independently. So each of these factors are independent of each other. They do not influence each other. Okay, two types of genetic crosses. We have a dihybrid cross, which we can see over here, and we have the monohybrid cross. The monohybrid cross is the one that you will be asked to draw or to complete. You wouldn't be asked to complete a dihybrid cross. You would only be given a dihybrid cross and asked questions based on it. But you should know how to complete that dihybrid cross in order for you to be able to understand how the answers came about, right? So with dihybrid crosses, you would have 16 offspring, and of these 16 offspring, they are usually going to give you the 9:3:3:1 ratio. If your original parents were two homologous individuals that are different, contrasting, and then you would use that to form these gametes here, and from there you would cross to get the offspring that we have, right? And then also here you would notice that when we may have started with round and yellow and wrinkled and green, but now we are going to now come up with two new characteristics, which is round and green and wrinkled and yellow. It takes us back to the law of independent assortment. Here, it doesn't mean that yellow seeds always have to be round. It doesn't mean that wrinkled seeds always have to be green, right? But the yellow has now matched itself or paired itself with wrinkled, and the green has now paired itself with round. So this basically shows us that law of independent assortment. Now, when we're looking at the monohybrid crosses, we are looking at only one characteristic at a time. With dihybrid, we are looking at 2 characteristics. Here we're looking at shape and color, whereas here we would look at shape only or color only, and here we would have been looking at tallness or height, right? We have tall and dwarf. And then this basically shows us again the law of dominance, whereby, remember, we said 2 contrasting homozygous organisms will lead to organisms that show, or offspring that show, the characteristic of the parent that is dominant. In this case, being tall is dominant over being dwarf. So we have crossed a tall homozygous individual with a dwarf homozygous individual. Remember, dwarfness here is recessive. The recessive characteristic can never be heterozygous. The recessive characteristic is always homozygous. Now, what do we see in our offspring? All the offspring are heterozygous tall. Okay. And then the types of dominance. We have 3 types of dominance. You should be able to differentiate these 3 types of dominance. We have complete dominance. With complete dominance, that is where you would see that you have a dominant allele and a recessive allele, right? Whereas with codominance and incomplete dominance, there is no recessive allele here. We only have dominant. They are codominant or they are incompletely dominant, but they are both equally dominant. Here, this R for red would be dominant over this R for white. Remember, the R for red, the letters we use here takes the letter of the dominant characteristic, R for red. We do not use W for white. White is recessive, therefore it takes the letter of the red. And then all the offsprings are going to be heterozygous red. The white will not show up because it's recessive. It's being masked by the dominant allele. Codominance and incomplete dominance. Red takes its letter, white takes its letter because they are both equally dominant. None of the characteristics are stronger than the other. So here we have RW, meaning that they are both present. They both appear in the offspring. Now, because they both appear in the offspring, you would only see with the difference in appearances to indicate whether this is codominance or incomplete dominance. With codominance, the RW is going to show up in the physical appearance as patches of red and white. As you can see, we can see a distinct red and a distinct white. Patches of red and white are codominant, right? They're coexisting. However, with incomplete dominance, we are going to see that a new color is going to show up. A new trait or new characteristic is going to show up. This new trait for us is pink. So RW here represents pink. Here, RW represents red and white. If you see a new color, that is incomplete dominance. If you see both colors appearing, that is codominance. Right. Now, when you are given a cross for complete dominance, you use 1 letter, a capital letter and a small letter. When you are given a question on codominance or incomplete dominance, you use 2 capital letters because both traits are dominant. Okay. And then we have sex-linked characteristics or sex-linked inheritance. Don't make the mistake of using the same annotations for the normal inheritance. Here we are looking at sex-linked, whereby we are looking at the genes that are located on the sex chromosomes, and that is why we say they are sex-linked genes, right? So here you would see that we are going to see the characteristic that we are studying being attached to the X chromosome. Now, in females, remember, we have XX, and in males, we have XY. So it means these characteristics will only be found on the X chromosome of the male, but will be found on both chromosomes of the female. So fathers can pass this allele to their female children. However, they will not pass the X-linked characteristic to their male offspring because fathers only pass the Y chromosome to their sons. So sons only inherit Y chromosomes from their father, meaning that if the father has this inheritance or this characteristic on their X chromosome, none of that male offsprings are going to receive that characteristic. So if it is a disease and it is on the X chromosome of the father, all his sons will not have that disease because they are not inheriting the disease. They are inheriting the Y, which does not carry the disease. However, all the females will then receive that disease, right? Because all the females would receive the X from their father. It is then going to be a different story when the disease is found on the X chromosomes of the mother, because the mother passes on the X chromosome to their son. Mothers only have X chromosomes, therefore they will pass that X chromosome that the son has. So mothers can pass the X-linked alleles to both sons and daughters, and the chances they are 50-50. Okay, so as you can see, we have the X chromosome, and 2 X chromosomes in the female, and there is that indication of the characteristic. In the male as well, there is that indication of the characteristic, but it is not found on the Y chromosome. So do not make the mistake of indicating X, H, Y, H. The Y does not have the H. It is XH, Y. But for females, it is XH, XH. Right. Okay. These sex-linked disorders or characteristics will then be seen in hemophilia and color blindness. So when you are asked a question on both color blindness and hemophilia, you know that it is a sex-linked characteristic. Therefore, you are going to use X to do your crosses. Then moving on to genetic lineages. Here we are looking at our pedigree diagrams. Remember, circles represent females, squares represent males. Shaded means that the individual is affected. Unshaded, it means the individual is unaffected. This line here between the two individuals show that they are reproducing. This indicates reproduction. This indicates the offspring. Now, these are all the offspring of these two individuals. Let's say individual 1 and 2, 3, 4, 5, and 6 are the offspring of individual 1 and 2. They are all siblings, right? Two individuals in this family are affected, 2 are unaffected. 2 are females and 2 are males. This is the first generation and this is the second generation. So if they are asking in which generation is the affected individual, you would say in the second generation. How many individuals in the second generation are affected? 2 individuals. How many males in the second generation are affected? 1 male is affected. How many females? 1 female is affected. What is the percentage of males that are affected in the second generation? 1 out of 3 males are affected. Therefore, you'll say 1 divide by 3 multiply by 100, and that will give you the percentage of those individuals that are affected. Now, let's look at the steps of solving questions on pedigree diagrams. I'm going to read this information and then the steps. It says here, the pedigree diagram below shows the pattern of inheritance of a certain genetic disorder controlled by a recessive allele. So this order is controlled by a recessive allele, right? The dominant allele is represented by N and the recessive is represented by a small letter n. It means that a person who has this genetic disorder would have to have 2 recessive alleles for the disorder to show up. All right. Now we know that these 2 individuals are affected. Because those 2 individuals are affected, we already know that they have 2 recessive alleles, right? First step, study the key. This is the key. This is also the key, right? We know normal, affected, right? Look for the dominant and the recessive characteristics. Already we know that having the disorder, you are recessive. Being normal, you are dominant, right? Fill in the genotype of the individuals with the recessive condition. Here, which individuals have the recessive condition? The ones that are affected, right? So the ones that are affected have the recessive condition. We will then fill in those 2 because we are sure about those. Now, each individual with the recessive condition inherited one allele from each parent, right? Obviously here it means that the parents have at least one recessive allele. This tells us that these parents are going to be like this. How do we know? Because it tells us that one parent gave the small letter, the other parent gave another small letter. Same applies here. But because these two parents are not affected, we also know that the parents have a capital letter N. Even with these two individuals, we know each of them have a capital letter N because it is the one that is going to mask the recessive allele for these individuals to be normal. This tells us that these parents are heterozygous because they were able to give their children the small letters, right? Okay. Then you work backwards and fill in one recessive allele for each parent, which we've already done here. We filled in the recessive allele, but we already also knew that they had the dominant alleles, right? If the parent has a dominant characteristic, fill in the capital letter as the second allele. We've done that already. Any other individual showing the dominant characteristic is most likely homozygous. So these individuals here can either be homozygous or heterozygous. So they can either be like this or like that. But whichever one they are going to be unaffected. If you have the third generation and the fourth generation, you will use that as a guide to determine whether these two individuals are homozygous or heterozygous. For example, let's look at this individual. If this individual has an affected offspring, it means that that offspring is small letter n, small letter n. For this offspring to be small letter n, both parents have to contain at least one small letter. So we would conclude that they are heterozygous. But if all their offspring are homozygous, then we can also conclude that this person is homozygous dominant. Okay, so it is just a matter of working backwards and forward to make sure that you fill in everything. But start with the recessive. Okay, then we have blood grouping. Here we are looking at blood grouping as an example of the inheritance of multiple alleles because it is controlled by 3 alleles, IA, IB, and I, right? And then these 3 alleles have different combinations that are going to lead to 4 blood groups, which are blood group A, B, AB, and blood group O, right? Blood group A and B are equally dominant. As we can see, they are represented by the capital letters, whereas blood group O is recessive. So if there is the crossing between blood group A and B, we now have an example of co-dominance. It's co-dominance because both of them are appearing here and we have IA, IB. So that is an example of co-dominance, right? But if we're looking at blood group A and blood group O, or blood group B and blood group O, that is complete dominance because A is dominant over I or B is dominant over the I that we see over there, right? Blood group A can be represented by 2 genotypes, IAIA, which is homozygous, or IAI, which is heterozygous. This gives us blood group A because I is recessive. Same applies with blood group B. But with blood group AB, both of them are going to appear. Because I is recessive, for a person to be blood group O, they need to have a genotype of the homozygous recessive. Now, we are going back again to the use of DNA profiles. We can use blood grouping and DNA profiling together to determine the father of a child, starting with the blood groups. So, by knowing the blood group of a child and the blood group of both parents, you are able to determine if the parents are the parents of that child. If the combination of blood groups do not allow those two people to be the parent of the child, it means that one of those parents is not the parent to the child, or the child does not belong to those parents, right? Now, if we have maybe 1 female and 4 males like this. And of these 4 males, their blood groups do allow them to be possible fathers to the child. Then you would further do that profile, that DNA profiling technique to confirm which one is the father. However, if based on the blood types, one of the males here do not qualify to be the father, we then do not even go on to have a DNA profile done on that person, because based on the blood group, they cannot be the parent. So blood groups are able to help us to show that a person is definitely not the father of the child or definitely not the mother of the child. However, it does not confirm that they are the parent. Just because the blood groups are matching, it doesn't mean they are the parent. Further tests have to be done because many other people out there have the same blood groups, and that also makes them the possible parent. Now let's have a look at an activity on sex-linked characteristic. Okay, it says Fragile X syndrome is a sex-linked dominant inheritance. You've been told that it is a sex-linked dominant inheritance. The allele that causes this condition is dominant. So a person who has the condition would have to have the dominant allele, while people without Fragile X have the recessive allele. Okay, we have some questions here, but before we get to the questions, remember, if a person has XR, they have Fragile X. If a person has XR, they still have Fragile X, because the dominant allele is going to mask not having the disorder. However, if the person has XR, they are not going to have that syndrome. Make sure that when you answer a question on sex-linked, you use the correct annotation, because if you say RR or RR, this is incorrect. You're not going to get marks because you've used the wrong annotation. Okay, just a few minutes to go through those 2 questions, 5 minutes for the 4-mark question, and then we will then discuss the questions. Okay, 2.5.1, state the phenotype of an individual with genotype X, R, capital letter, and X, small letter r. I've just mentioned that just now. Okay, genotype and phenotype. Know the keywords. Genotype, it's the letters. Phenotype, in words, the physical appearance. This person has the Fragile X syndrome characteristic because the R is there, right? So female, again, you must mention that this is a female. How do you know? Because it's 2 X's, right? So it's a female with Fragile X syndrome. If the question was X, R, Y, you would say this is a male with Fragile X. If it was X, small letter R, Y, a male without Fragile X. Okay. 2.5.2, explain why both males and females have equal chances of suffering from Fragile X. Now, because this is a dominant disorder, or this is a disorder that is caused by a dominant allele, the chances of having the disorder are going to be equal. So it is caused by a dominant allele on the X chromosome. So both the males and the female only need one to have the disorder. Like I said, you only need X capital letter R to have the disorder. So if you have X capital letter R, you will have the disorder. Either X capital letter R, X small letter r, or X capital letter R, Y. The chances are still the same. Okay. And then 2.5.3, this is a genetic cross. I'm going to give you about 7 minutes to go through the question. Make sure that you use the correct format to complete your cross, right? And then we will have a look at that. All right, so here, remember, it says a man with the genotype XRY marries a woman with the genotype XR small letter and XR small letter. So a female without Fragile syndrome and a male with Fragile syndrome. Use a genetic cross to explain the probability of this couple having a son with Fragile X. Remember, correct format. You start with P1, meiosis, fertilization, F1, and then the phenotype of the P1, the genotype of the P1. Phenotype in words, genotype in letters, right? They said the female is homozygous recessive, the male is dominant, right? And then you're going to have your meiosis leading to the gametes, separate your gametes, right? And then do the cross, that's fertilization. You cross, you have your offspring, the genotypes of the F1, which are the offspring. This one is a female with the disorder, another female with the disorder, a male without the disorder, and a male without the disorder. By simply looking at the genotype of the mother, you should already know the genotype of the son. How? This female has no disorder. She's going to pass any of these two alleles to the son, right? That means that the son is not going to have the disorder, because we're not even considering the Y, because the Y does not come with the allele, right? So the males are both unaffected. So 2 males without Fragile X, meaning that the percentage chance, the probability of the son having Fragile X is 0. No chances. So before you even do your cross, you must already know the answer here. Okay, let's move on to evolution. Right. So here we are going to be looking at the biological evolution, which means that all present life forms are the descendants and they are related to those that lived in the past. They are descendants of those that lived in the past, and they are actually related to those that lived in the past, right? This also tells us that although we may look different because of the modifications that have taken place over time, we are still related. So there are going to be some changes, which we refer to as modification by descent. Remember, as the descendants come, there are going to be some modifications or some changes, which is why we are now able to see these changes that we are looking at here. This individual or this organism is related to this one, and this one is a descendant. However, we can see the modifications with time. So they have been modified from one generation to the next. Right. Okay. So from here, we will then look at the evidence of evolution. Here we will be looking at some of the pieces of evidence that actually show us or prove to us that we have descended from those that lived in the past, and also that we are related. So we have 4 ways of showing this evidence. We have homologous structures, whereby here we are looking at the fact that although these 4 organisms here may have different structures in appearance and different functions, we can still see that they have the same basic structure, meaning that we can see all of them having the humerus. They all both have the radius and ulna. They all have the carpals, the metacarpals, as well as the phalanges. However, these 4 organisms are different, and they use these limbs for different functions. Because, for example, humans use this for holding, cats use this for walking, the whales for swimming, the birds for flying. So different functions, but the same basic structure, which shows us that there is some relationship there. Then we may also have fossil evidence, which has been gathered by different individuals that have gathered this and looked at some of the similarities and differences between the modern day humans and those that lived millions of years ago, and they are able to find some of those similarities that then show us that we are indeed related. We also have biogeography, which shows different organisms from different areas and the relationship between those based on some of the structures that may be different or show some similarities. And then lastly, we have genetic evidence, whereby the genetic material is compared to see how closely related certain organisms are to each other. And then we also have the sources of variation, right? So here we are going to be looking at things such as the random fertilization. Because fertilization takes place randomly, we do not know what kind of ovum is going to be fertilized by what kind of sperm cell. So this is random. It shows that it contributes to genetic variation. Genetic variation is very important because when organisms of the same species are different, organisms of the same species show a great deal of variation. It means that this increases their chances of survival should there be any changes in the environmental conditions. Some organisms will survive while some organisms will not survive. So because of random fertilization, we can have genetic variation, and that will lead to that survival. Okay, we also have mutations. So as some mutations occur, we are going to now have differences in the organisms of the same species. Those differences are what will now make these two organisms different, show genetic variation. Therefore, they are going to also react to the changes in the environmental conditions differently. Next, we have meiosis. Under meiosis, we look at crossing over and random arrangement of chromosomes at the equator. So here we look at crossing over, which is what we are seeing here, as well as the random arrangement of chromosomes at the equator. Remember, because these chromosomes are looking like this, and at the end of the process of crossing over, they are now looking different. It means that the cells that are going to be produced at the end of meiosis are going to be completely different from each other. Adding to that, random arrangement. The chromosomes are arranged randomly. Therefore, we do not know which chromosome is going to form part of which cell. That leads to the cells being different from each other. And lastly, we have random mating. When organisms undergo mating, we don't know which organism is going to mate with which organism, meaning that we do not know which genetic material is going to combine with which genetic material. This is random. Therefore, this leads to a great deal of variation within organisms of the same species. And then we also have the two types of variation. Here we have continuous variation and discontinuous variation. We can see there's a continuation here. Straightforward, there is a continuation. We have this very short person, and then the height keeps increasing until we have a very tall person. So this is a continuous thing. Whereas with discontinuous, there isn't a range of different characteristics. You can have this or that, nothing in between. You can have a male or a female, nothing in between. You can have brown or blue eyes. There isn't any in between the blue and the brown. Blood groups as well, there isn't in between. The ability to roll your tongue, you either roll your tongue or you don't. There isn't anything in between the rolling and being unable to roll. Now, looking at the theories of evolution. Here, we look at Lamarckism as well as Darwinism, meaning that we are going to look at the explanations that were made by Lamarck on the evolution of organisms, as well as how Darwin also explains evolution of these different organisms. Okay, now let's have a look at what Lamarck says. So Lamarck explained how animals changed over time by what he called the two laws of use and disuse. Those are his two laws, right? The law of use and disuse, and the law of the inheritance of acquired or modified characteristics. The first law, remember, he talks about how if an organism or if an animal uses an organ more, that organ will be better modified and it will become bigger and stronger and be able to perform the function more. However, if the part or the organ is not used, that organ will become smaller and smaller until it disappears. Right. So the law of use, The more you use it, the more it grows and becomes better. The less you use it, the more it shrinks and eventually disappears. So, for example, he says that the giraffes often stretched their necks to reach for the leaves on the top of the trees, and as they were stretching their necks became longer and longer. Hence, we now have giraffes with long necks, right? So he also mentioned how the snakes used their legs less and eventually their legs disappeared, right? Now, the law of inheritance of acquired characteristics speaks about how these acquired characteristics, this tall neck, is now going to be passed on to the next generation, right? So these laws were rejected, and they were rejected because there is no evidence that the acquired characteristics can be passed on, because the change in your phenotype is not a change in your genotype, right? And also, there is no genetic basis to support what Lamarck was saying. However, Darwin mentions or talks about the evolution by natural selection, right? Whereby he talks about that there is a great deal of variation in the appearance of organisms or in their ability, and during reproduction, there is a large number of offspring. These large number of offspring will show that great deal of variation, right? But some of them will not survive, while some will survive. Why? Because there is going to be competition. They are going to compete for the limited resources, whereby the ones with the good characteristics or the favorable characteristics will survive, whilst the ones with the unfavorable characteristics will not survive. Those with the favorable characteristics will then reproduce, and from one generation to the next, the ones with the favorable characteristics will continue to increase, while the ones with the unfavorable characteristics will continue to die and eventually will disappear, right? So the genetics are being passed on to their offspring, and we will now say nature has selected the organisms with the favorable characteristics to survive. So we call that natural selection, right? When Darwin speaks about this, he also talks about how this process takes place gradually, right? So we have gradualism, whereby it is characterized by long periods of time. This gradualism is characterized by small changes. As we can see, the changes are taking place slowly. We started off with orange. The color is slowly changing until we have white. Here also, the color is slowly changing until we have this dark red. However, with punctuated equilibrium, we look at long periods of time of no change that are characterized by, that alternate with short periods of time of rapid change and big changes. As we can see, we started off with the orange, and over a very short period of time, we suddenly have white. And over a short period of time, we suddenly have the dark red. And then long periods of time with no changes. So punctuated equilibrium is characterized by a long period of time of no change and short period of time with very quick and rapid changes. So according to Darwin's theory, evolution occurs gradually. So this is gradualism, and it takes place over a long period of time. Okay. And then you should also be able to differentiate between artificial selection and natural selection. We're not going to look at that a lot, but you should know that with artificial selection, humans are mostly involved, whereas with natural selection, nature would be the driving force of that. Then let's move on to speciation. Remember, here we're looking at the formation of new species. Know these three definitions: the population, the species, and the speciation. So remember, here we would usually start off with one population. That population will be separated into two species, and it can either be allopatric speciation, whereby it is by a geographical barrier, or sympatric speciation due to reproductive isolation mechanisms. Right. Okay. So if it is allopatric, you should know the type of geographical barrier, which is what we are looking at here. Right. Here you should be able to describe speciation as it is here. You should know that the geographical barrier, you're going to name it. And then from there, you should mention that there is no gene flow between the two populations that have been separated by this barrier, right? Because there's no gene flow, it means that now these two populations are experiencing different environmental conditions. So natural selection is taking place independently. Because it's taking place independently, the two populations are going to be genotypically and phenotypically different from each other. And they are not able to interbreed. Therefore, even if they were to meet again, they will not be able to interbreed. How do we see that the organisms are not of the same species? We will see that we're going to have an infertile offspring, which takes us to the reproductive isolating mechanisms. We have 5 of these, which is the breeding at different times of the year, species-specific courtship behavior, adaptation to different pollinators, infertile offspring, and the prevention of fertilization. Okay, also go through the evolution in present times. Here you are going to look at how evolution has affected certain things that are happening in present times. We can look at the use of DDT, how DDT, mosquitoes have become resistant to the DDT, how different things can become independent to certain medications due to the changes, the adaptation. And as the changes are occurring, you would end up having this resistant types to whatever medicine you would be using, right? And then you should also be able to interpret a phylogenetic tree. So you should know that this is where we have the ancestor, and then as it branches, that shows that there are the descendants. And you should also be able to know that if we are looking at organisms here, and for example, A. robustus and the Homo erectus and the Homo habilis, you should know which ones are more closely related to each other. And also the time. Look at the time frame, and it will tell you when the speciation has taken place, when the extinction has taken place, and so on. Okay, now looking at human evolution, the characteristics humans share with African apes. You should know all of these, know how the large brain similarities are, the being able to see organisms or things in front of us, the ability to rotate the arms, the upright posture, and the flat nails instead of claws. So those are some of the similarities. So have a look at the similarities and just remind yourselves of those. And then we also have the differences. What are the differences between Homo sapiens and other primates? Have a look at that from the brain size, the prognathous jaws, the flat face versus the face, the head that slopes, the position of the foramen magnum, which is very important, and the size of the canine and the shape of the teeth in the mouth, right? And the chin. We have there's a well-developed chin and that poorly developed chin. Those are the differences that you should know. And then the shape of the spine as well. As you can see, the spine will be different. The foramen magnum, the forwardly positioned foramen magnum and the backward. Also, the pelvic girdle, the short and broad and the tall and narrow. You should be able to identify all of these differences. And lastly, looking at the out of Africa hypothesis. Remember here, the idea is that all organisms originated from Africa and they migrated to all the other parts of the world. Hence, they say it is the out of Africa hypothesis. Here, you focus on the evidence. What evidence do we have that these species moved out of Africa? You're going to mention the fact that some of the old fossils are found in Africa and in Africa only. Therefore, it means that they originated here. And then the old ones are found in Africa and other parts of the world. Because the old one is found in Africa, it means that the old one was here and the younger ones moved to the rest of Africa. So you should know that the oldest fossils that are found would be those of Homo ergaster as well as the Homo sapiens. You should also be able to give genetic evidence in genetic evidence, remember, we look at the mitochondrial DNA, we look at the mitochondrial DNA that we found in females that is only going to be passed on from one female organism to the next. The mutations in that are an indication of the out of Africa hypothesis. Okay, let's have a look at our last activity. It says Littlefoot is a hominid fossil that was discovered at the Sterkfontein caves by Professor Ron Clark. As you go through this, you should also know the people who discovered the fossils. You should also know where the fossils were discovered. The bones of Littlefoot showed that this fossil had many characteristics similar to those found in humans. So as we can see, we are looking at the hand, the pelvic girdle, the skull, together with the foramen magnum and the shape of the teeth. Right. So just a few minutes, go through the questions so that we can be able to discuss them. (3.4.1) What is the scientific name for Littlefoot? So you should also know the scientific names. So Littlefoot is the Australopithecus africanus. And then 3.4.2, write down the letter of the structure suggesting this genus. So you should then be able to look at the diagram that we were given and identify the diagram that represents that it had the small canines and the fact that the spinal column entered the skull vertically. Bipedalism. Remember, when you look at bipedalism, we're looking at the position of the foramen magnum and that it was in a more forward position. And then the fact that it had similar forelimbs with the humans. It means that we are looking at the hands there. I think that is something that is straightforward. Obviously, if we are looking at the canines, here we do not see those big canines showing on the outside, meaning that C would represent A on our question there. And then the foramen magnum would be part D, entering more vertically, and then similar forearms as humans, it would be A. All right, that would be our answers for those questions, the C, D, and A. All right, describe how the shape of the structure B. Let's go back and see what structure B is. The structure of B, which is the pelvic girdle. Describe how the structure of the pelvic girdle shows this genus is more evolved than the primitive apes, but less evolved than humans. So here you're going to talk about how you should know how the humans have a short and wide pelvic girdle, whereas the primitive apes had a long and narrow pelvic girdle. But if you're looking at that pelvic girdle, it is somehow of a transitional fossil, which is somehow in between the two, right? So shape B is shorter and wider than that of primitive apes, but it is slightly taller and narrower compared to that of humans. So it does not give the exact shape of the pelvis of humans, but it is also not the shape of the primitive apes. So like I said, it is that fossil that is found in between. Okay, 3.4.4. Explain the significance of the change in the cranium size of Littlefoot to modern humans. So the cranium size. Remember, when we're talking about the cranium size, we're going to look at the fact that in humans, the cranium size is bigger. Therefore, bigger brains, meaning more intelligence. So the cranium size increased in modern humans to allow for more intelligence or to process more information. Right. And then the next one. Littlefoot is used as fossil evidence in the out of Africa hypothesis that we just looked at. A, name one line of evidence for out of Africa hypothesis. Remember, here we can use fossil evidence, we can use genetic evidence as well. So any that you can mention. And then B, explain how the fossils of Littlefoot genus are used in the out of Africa hypothesis. Obviously, if we're talking about the fossils, it means that we're looking at fossil evidence. Now, fossils of the Australopithecus were found in Africa only. The fact that the fossils of Australopithecus were found in Africa only is proof or is evidence that these originated in Africa and the youngest ones were found in other parts of the world. However, with these ones, they are not even found in other parts of the world. Therefore, it shows that they originated in Africa because they were not found anywhere else. Right, so it suggests that ancestors of Homo sapiens originated in Africa. Okay, and then our last question is describe how a new species forms from a geographical area. Right, remember I said you must be able to explain from the barrier until the fact that they are genotypically and phenotypically different from each other. And therefore, even if they were to meet, they will not be able to interbreed because they are now two different species. So if a population of a single species becomes separated by a geographical barrier, which can be any of those, in this case, because the question is general, you do not have to mention the barrier. However, if you are given a diagram and you can see the barrier, you must mention what is the barrier. If you don't, you will lose that mark. Then the population splits into two. There is no gene flow between these two populations since the population may be exposed to different environmental conditions. So the selective pressure is different. Natural selection is taking place independently. Natural selection occurs independently in each of those two populations, such that the individuals are very different from each other, both genotypically and phenotypically. Even if these two populations were to mix again, they will not be able to interbreed because they are now two different species. All right, so as we have looked at the 4 chapters that make up our paper 2, just go back and revise, have a look at what we've just discussed now. Make sure that you go through, especially with evolution, the evidence for evolution and the out of Africa hypothesis. Make sure that you know what exactly happens there. Know which species were found in Africa only, which ones were the oldest that were found in Africa, and so on. And like I said, know where they were discovered and who discovered these. Okay, so another reminder, make sure you bring all your mathematical instruments, your pen, your pencil, and your eraser into the examination. We are going to be having calculations such as your percentage increase, percentage decrease, calculation of percentages, calculations of the segments. If you are expected to draw a pie chart, always have those segments calculated before you draw your pie chart. Use the correct instruments for your pie chart, heading on your graphs and your tables and drawings. So make sure you bring everything that is going to be needed there. There's definitely going to be calculations, graphs, and other drawings. So make sure you come prepared. All right, we have come to the end of our lesson. Thank you for watching. Goodbye.